I want you to think about something you do, like dozens of times a day. Maybe you're reaching out right now to grab a cup of coffee. Or maybe you're doing something incredibly precise, like threading a needle.
Yeah, something we take entirely for granted.
Right. You have the thought, I want to do this, and instantly your hand just complies. It feels completely seamless.
It feels like magic, mostly because we just don't see the biological infrastructure underneath it all.
Exactly. But how does an abstract, electrical impulse in your brain actually turn into physical, mechanical force in the real world?
Well, beneath the surface, every single intentional movement requires this staggeringly complex sequence of events.
Yeah.
And they all execute perfectly in roughly 1 millisecond.
Which is mind-blowing. So today's deep dive is into a stack of anatomy and physiology notes focused entirely on that exact process.
Yes, our mission today is to explore that microscopic choke point where intention actually turns into action.
Right, the neuromuscular junction, or NMJ. And right off the bat, we need to shatter a massive misconception about human anatomy.
Oh, absolutely. The wiring misconception.
Yeah, when you think of nerves controlling muscles, you probably picture a copper wire permanently soldered to a motor. But, a nerve and a muscle do not touch. They never touch.
Right. There is a physical, fluid-filled void between them.
And that gap changes everything, doesn't it?
It really does. You have an electrical signal traveling down the nerve at high speed, and then suddenly the road just ends.
It just hits a wall.
Basically, yeah.
Yeah.
That signal has to somehow cross a physical void, translate itself into a chemical message, and then transform back into an electrical signal on the muscle side.
Just to trigger a mechanical contraction.
Exactly. And if any part of that translation fails, the movement fails. You are completely paralyzed.
Okay, let's unpack this, because for the longest time, the brightest minds in science didn't even know this gap existed at all.
No, they didn't.
Yeah.
19th century physiologists completely bought into that soldered wire idea you mentioned.
Right. They thought nerves and muscles were essentially fused, continuous tissue. Or they thought the electrical signal just, hopped over the gap like a literal spark jumping between two wires.
Yeah, they called it the sparks theory.
Mm-hmm.
And it made intuitive sense based on what they knew about electricity at the time.
Sure, it seems logical.
But the unraveling of that mechanical view actually started way back in the 1850s with a French physiologist named Claude Bernard.
And he wasn't even studying electricity, was he?
No, not at all. He was studying a poison, specifically curare.
Curare. So, the paralytic plant toxin historically used by South American hunters on their blow darts?
That's the one.
A paralyzing blow dart poison is a pretty dramatic way to start a physiological revolution.
It is, but his experiment was just so elegant. He takes a frog's leg, applies the curare, and then tests the individual components.
Okay.
He shocks the muscle directly, and the muscle contracts. Then he shocks the nerve directly, and the nerve successfully conducts the electrical impulse.
So both components are perfectly healthy.
Right. But when he stimulates the nerve to try and make the muscle move, absolutely nothing happens.
Wow. So the bridge was just out.
Exactly. The nerve was firing, the muscle was capable of receiving, but something specifically wedged in between them had been chemically blocked by the curare.
Which provided the first rigorous proof that the nerve and muscle were not just one continuous wire.
Right. There had to be a distinct, druggable junction separating them.
That is wild. And it took decades for the rest of science to catch up to that idea. Around 1905, John Newport Langley at Cambridge starts sketching out this concept of a receptive substance on the muscle.
Yeah, he was predicting molecular receptors decades before anyone had the microscopes to actually see them.
But the definitive proof of how the signal crosses the gap, the final nail in the coffin for the sparks theory, is honestly one of the wildest origin stories in modern science.
Oh, the Otto Lowy story.
Yes. In 1921, it all comes down to him.
Right. Lowy was agonizing over this debate. He was thinking, if the transmission wasn't an electrical spark, was it a chemical soup?
And the decisive experiment to prove this literally came to him in a dream.
It did. He wakes up in the middle of the night, scribbles down the experimental design on a tiny notepad, goes back to sleep.
And wakes up the next morning completely unable to decipher his own handwriting.
Yes. Imagine that. You solve one of the greatest mysteries of human physiology. You hold the secret to how the body moves, and your own messy handwriting ruins it.
That is so painful. But what's fascinating here is that his subconscious mind didn't let it go. The dream recurred the very next night.
Thankfully, yeah. And this time, Lowy didn't risk the notepad. He got out of bed at 3 in the morning, went straight to his laboratory, and performed the experiment on the spot.
The notes detail what he did, and it is just brilliant. He takes a beating, isolated frog heart. He stimulates its vagus nerve, which slows the heartbeat down.
Right, which makes sense.
And if the Sparks theory was right, that was just electrical current doing the work. But then he takes the saline fluid that this first heart was bathing in, extracts it, and just pours it over a totally separate, isolated frog heart.
And that second heart immediately slows down too.
Even though its nerve was never touched.
Exactly. The electrical current couldn't have transferred through the fluid like that.
So the first nerve didn't just fire electricity, it had secreted a physical chemical substance into the bath.
And Lowy called it vagus stuff. He proved that the nervous system uses chemical soup, not electrical sparks.
That is incredible.
And later, working in Britain, Sir Henry Hallett Dale proved that vagus stuff was actually a specific molecule called acetylcholine.
Acetylcholine, right.
Yeah, and Dale took it a step further and proved this exact same chemical is what crosses the gap at our voluntary skeletal muscles, the neuromuscular junction.
And they shared the Nobel Prize for this discovery, which is well-deserved.
Oh, definitely.
But, knowing that the gap exists and uses chemical soup is one thing. I want to dive into the actual mechanics now, because the sheer physics of what happens in that fraction of a millisecond when you decide to pick up your coffee is staggering.
It really is a marvel of biology.
Right. So the process starts when the electrical signal, the action potential, races down the nerve and arrives at the very end, called the axon terminal.
Yeah, and this terminal rests in a microscopic trough on the muscle fiber called a synaptic gutter.
Okay, so they're nestled together but not touching.
Correct. This is where we hit the biological choke point. The electrical wave can't cross the physical void of the synaptic cleft.
It has to trigger a chemical release.
Exactly. When that electrical wave hits the terminal, it actually changes the electrical charge of the cell membrane, which causes specialized gates to just fly open.
And these are the voltage-gated calcium channels, right?
Yes, voltage-gated calcium channels.
Okay, so I want to use an analogy here to make this relatable. Think of an army standing on one side of a river. They need to signal the other side to attack, but they can't shout, and they have no radios.
Okay, I like where this is going.
So they coordinate to throw thousands of water balloons filled with secret messages across the river all at once. The river is our synaptic gap. The electrical signal arriving is the commander giving the order. And the calcium rushing into the nerve terminal is the commander yelling, "Throw the balloons!"
That's a great way to visualize it. Because that calcium influx is the non-negotiable trigger for the entire system.
How so?
Well, nature relies on concentration gradients. Calcium ions are highly concentrated outside the nerve cell and incredibly scarce inside.
Oh, I see. So it wants to get in.
Exactly. When those voltage gates open, the calcium violently rushes in, driven by the desire to equalize that pressure.
And once inside, that calcium interacts with the nerve's internal storage system.
Right. Your water balloons. We call them synaptic vesicles.
Synaptic vesicles.
Yeah. These are tiny spherical membrane-bound sacs clustered right at the edge of the nerve terminal. And each single vesicle is preloaded with roughly 5,000 to 10,000 molecules of acetylcholine.
Wow. In just one vesicle.
Just one. And when the internal calcium spikes, it forces 100 to 300 of these vesicles to hurl themselves against the cell membrane.
They fuse with it, burst open, and just dump their massive acetylcholine payload directly into the synaptic cleft.
Exactly.
So we are talking about millions of individual molecules being dumped into this microscopic river all at once.
Yes, and this gap is incredibly small, only about 50 to 100 nanometers wide.
Which is nothing.
Right, so the acetylcholine diffuses across that void in microseconds, and waiting for them on the muscle side, the motor end plate, are the targets.
The nicotinic acetylcholine receptors.
Precisely. And the architecture of that motor end plate is heavily optimized to catch those molecules.
Because the muscle membrane isn't flat, right?
No, it's folded into these deep microscopic trenches called junctional folds. This vastly increases the surface area.
And clustered heavily at the crests of those folds are the receptors.
Packed at a density of up to 10,000 receptors per square micrometer.
That's insane. So this is where the translation happens. The chemical message hits the receptors.
Right. And when two acetylcholine molecules bind to a single receptor, it physically changes shape. It twists and opens up a microscopic tunnel right through the muscle membrane.
And just like calcium was waiting outside the nerve, sodium is waiting outside the muscle.
Exactly. When those tunnels open, sodium violently rushes into the muscle cell.
And this massive influx of positive charge creates a localized electrical spark.
Yes, called an end plate potential, or EPP.
So the chemical signal has officially been translated back into an electrical signal.
Right. And if that end plate potential is large enough, it hits a threshold and triggers a full-blown muscle action potential. The electricity is reborn.
Wow.
It races along the surface of the muscle fiber, but it also has to get deep inside the muscle tissue to make the whole thing contract simultaneously.
Right, because a muscle fiber is thick.
Exactly. So it does this by diving into a network of internal tunnels called T-tubules.
So the chemical balloons hit their targets, the gates open, and a massive electrical siren goes off across the entire surface of the base, eventually echoing down into the deep underground barracks.
That's it. And those T-tubules carry the electrical wave deep into the core of the muscle fiber, where it meets a structure called the sarcoplasmic reticulum.
Which is like an internal storage tank entirely filled with calcium.
Yes, more calcium. The electrical signal forces that tank to dump its calcium into the muscle fiber's main body.
And that internal flood of calcium is the final key.
It is. It binds to the muscle's contractile proteins, actin and myosin, allowing them to physically lock together and pull.
And the muscle physically shortens.
Exactly. That is excitation-contraction coupling.
It is a brilliant cascade. But, reading the notes, there's a glaring problem if the process stops there.
Oh, absolutely.
If those acetylcholine molecules just stay locked onto the receptors, the sodium keeps rushing in, the siren keeps blaring, and the muscle stays permanently locked in a rigid cramp.
Right. The army on the other side of the river would just be deafened by the constant signal.
We need a cleanup crew.
We do. The
Yeah.
system requires an instant reset mechanism, which comes in the form of an enzyme called acetylcholinesterase.
Acetylcholinesterase.
Yes, it's permanently stationed right in the synaptic cleft, anchored to the extracellular matrix. Its sole purpose in life is to find acetylcholine and instantly shred it into two harmless pieces, acetate and choline.
So it literally vaporizes the chemical message the millisecond it's received.
Exactly. It pulls the acetylcholine off the receptors,
Yeah.
the tunnel snaps shut, the electrical siren stops, the internal calcium is pumped back into the storage tanks, and the muscle relaxes.
Entirely primed and ready for the next command.
Yes. And from the nerve impulse arriving to the acetylcholine crossing the gap to the muscle twitching and resetting, the entire cycle consumes just a few thousandths of a second.
Now here's where it gets really interesting. This multi-step chemical relay race, relying on millions of molecules diffusing across a fluid gap perfectly, is required every single time you twitch a finger. It seems so statistically precarious. If it's this incredibly complex, why doesn't it fail more often? Like, why aren't we just randomly dropping our coffee cups mid-sip because a few vesicles didn't fuse?
It's a great question. And it's because evolution built an extreme fail-safe into the architecture. It's called the safety factor.
The safety factor.
Right. The body does not operate on the bare minimum required for transmission. A single nerve impulse typically releases enough acetylcholine to produce an end plate potential that is 3 to 5 times larger than what is actually needed to hit the threshold.
3 to 5 times. So we are wildly over-delivering the chemical payload.
Oh, absolutely. You only need a spark to start the engine, but the body is throwing a stick of dynamite at it just to be absolutely certain the muscle fires.
That's wild. And the mechanics of how that dynamite is packaged brings us to the pioneering work of Sir Bernard Katz in the 1950s, right?
Yes, Katz. He used intracellular microelectrodes to record the faint electrical activity right at the motor end plate of resting muscles. And he discovered what we now call quantal release. He proved that acetylcholine isn't just leaking out in a smooth, continuous stream like water from a faucet.
Right.
It is always released in fixed-size packets, or quanta.
And those packets correspond exactly to the individual synaptic vesicles, the water balloon.
Exactly. Every balloon holds the exact same amount of chemical.
But reading about Katz's experiments, I found something that completely broke my mental model of how still and quiet the body is at rest.
Oh, the MEPPs.
Yes. The notes talk about miniature end plate potentials, or MEPPs. Katz found these spontaneous 0.4 millivolt electrical blips happening constantly, even when the nerve was entirely dead silent.
Right, no electrical signal at all.
Yeah. If there is no electrical command coming from the brain, why is my junction firing off these tiny blips? Are my nerves just accidentally leaking neurotransmitters?
Basically, yes.
Yeah.
It's the thermodynamic reality of molecules existing in a fluid environment.
Oh, interesting.
Even with zero electrical command and no massive calcium rush, a single vesicle will occasionally just bump into the cell membrane, by sheer random thermal motion.
Just floating around and bump.
Exactly. It fuses and dumps its single payload of acetylcholine into the gap. That one packet crosses over, opens a few receptors, and causes a tiny 0.4 millivolt blip on the muscle, the MEPP.
So it's just background biological noise?
It is. And because of that threshold requirement we talked about, that tiny 0.4 millivolt blip isn't nearly enough to trigger a full muscle contraction.
So you don't randomly spasm?
Right. But it is direct physical evidence of the probabilistic nature of the synapse.
Wow.
A deliberate, powerful muscle contraction is really just the synchronization of hundreds of these random, accidental-style vesicle fusions forced to happen, all at the exact same millisecond by that sudden flood of internal calcium.
That completely shifts the perspective. The neuromuscular junction isn't this rigid mechanical switch. It's a dynamic, slightly leaky, statistically governed chemical cloud. But if this exact same leaky, probability-driven chemical dump is happening everywhere, how does the body scale it to perform completely different tasks?
What do you mean?
Well, the precision required to thread a needle is fundamentally different from the raw power required to deadlift a barbell, right?
Ah, yes. The biological solution to that is the motor unit.
Okay.
A motor unit is the fundamental building block of movement.
Mm-hmm.
It is defined as one single motor neuron and every individual muscle fiber that it directly controls.
Which dispels another major misconception. People just assume one nerve fiber goes to one muscle fiber, a perfect one-to-one ratio across the board.
Right, but that's not how it works. One neuron actually branches out at its terminal end to innervate multiple muscle fibers.
Okay.
And the specific ratio of one neuron to the number of muscle fibers it commands dictates how your body trades off precision against power.
Oh, wow. And this wiring is highly plastic, right? It can remodel itself over time.
It can.
So my eye muscles have a completely different wiring ratio than my calf muscles.
Vastly different.
Yeah.
Look at the extraocular muscles that aim your eyes. When you are reading a book or tracking a fast-moving object, you need exquisite microscopic adjustment.
Right.
To achieve that, the motor units are incredibly small. One single motor neuron might control only 10 to 20 individual muscle fibers.
Oh, wow.
So when that neuron fires, only a tiny granular fraction of the muscle engages. You have phenomenal fine-tuning capabilities.
But if I look at a massive muscle built for locomotion like the gastrocnemius in my calf...
Right, you don't need microscopic fine-tuning to propel your body weight off the ground. You need explosive force.
Yeah.
In the calf muscle, a single motor neuron might branch out to control well over 1,000 muscle fibers simultaneously.
No, 1,000.
Yes. When that one brain signal comes down the pipe, 1,000 fibers contract at the exact same millisecond.
Wow.
You generate raw, efficient power, but you entirely sacrifice the ability to delicately scale the force.
So you could never paint a masterpiece or thread a needle using your calf muscles?
Exactly. The limitation isn't in your brain. It is literally hardwired into the mathematics of the motor units.
That is fascinating. Now, if we connect this to the bigger picture, you start to realize how profoundly vulnerable this system is.
Oh, it is the ultimate Achilles heel.
Right, because the entire human experience of movement relies on this hyper-specific chemical bridge. It depends on a precise sequence of voltage gates, calcium rushes, acetylcholine, and cleanup enzymes.
It represents the ultimate physiological choke point.
And because of that, it is one of the most heavily weaponized and medicalized structures in the natural world. If you can disrupt the gap, you control the host.
Absolutely.
Let's look at what happens when this bridge is attacked. Based on the notes, there are two catastrophic failure modes. Mode 1 is the signal never arrives.
Okay, think back to Claude Bernard's experiment with Cure.
The blow dart poison.
Right. Cure is a competitive antagonist. Its molecular shape is just similar enough to acetylcholine that it fits perfectly into the nicotinic receptors on the muscle.
Oh, so it mimics it.
Sort of, but it doesn't open the tunnel. It just sits there, physically blocking the keyhole.
So the nerve dumps the acetylcholine, but the chemical just bounces off.
Exactly. The message never lands, resulting in immediate flaccid paralysis.
The muscles go completely limp because they are deaf to the command.
Right.
But then you have something like botulinum toxin produced by the bacteria Clostridium botulinum. This does something even more sinister, right?
It does. It doesn't block the receiving end. It sabotages the launch pad.
Oh, wow.
The mechanism of botulinum toxin is a masterclass in biological warfare. Inside the nerve terminal, for those water balloons, the vesicles to fuse with the membrane, they rely on a complex of proteins called snare proteins.
Okay, snare proteins.
You can think of snare proteins as a molecular zipper that docks the vesicle to the cell wall and pulls it tight enough to fuse.
And the toxin.
Botulinum toxin acts as a pair of chemical scissors. It gets inside the nerve and physically snips those snare proteins in half.
So the zipper is broken, the commander yells fire, the calcium rushes in, but the vesicles just bounce against the membrane.
Exactly. They can't dock, they can't fuse, and the acetylcholine is permanently trapped inside the nerve.
So the nerve fires perfectly, but absolutely no chemical crosses the void.
Nothing. This makes botulinum toxin one of the most lethal, potent neurotoxins ever discovered by humanity.
Really?
Yes, a microscopic amount can paralyze the respiratory muscles, leading to asphyxiation.
And yet, this highlights one of the most surreal contradictions in modern medicine, doesn't it?
Oh, for sure. We take this exact same lethal mechanism, heavily dilute it, dose it in minute quantities, and inject it clinically.
We call it Botox.
Yes.
Botox.
We use the deadliest known toxin to intentionally paralyze facial muscles to smooth out wrinkles or to medically break the bridge in patients suffering from chronic migraines or severe muscle spasticity.
Right, we are deliberately weaponizing the choke point for relief. It is a phenomenal example of the dose making the poison. That is so wild. But that's only mode 1. Mode 2 is the exact opposite nightmare. The signal crosses the gap, but it never turns off.
Yes.
This goes back to the cleanup crew. Acetylcholine doesn't just naturally drift away or wear off. It has to be actively, violently destroyed by acetylcholinesterase.
Right. And if you inhibit that specific cleanup enzyme, the entire system collapses into chaos.
Which is exactly how organophosphates operate.
Yes. These chemical compounds are the active ingredients in many agricultural pesticides, and they form the backbone of military nerve agents like sarin gas.
Oh, wow.
They chemically bind to acetylcholinesterase and permanently disable it.
So the acetylcholine is dumped into the gap, it hits the receptors, the sodium rushes in, the siren goes off, and then the chemical just sits there, locking the dates open.
And the chemical message is never cleared. The muscle receptors are stimulated continuously, hundreds of times a second.
That sounds horrible.
It is. The muscle is locked in a state of sustained, agonizing overstimulation called tetany. It manifests as violent spasms, and eventually, this leads to paralysis as well, but by a completely different route.
So it is paralysis by sheer exhaustion and rigid contraction rather than the limp, flaccid paralysis of Curare.
Exactly.
It's terrifying how tweaking one microscopic enzyme completely hijacks the human body. But the attacks don't just come from jungle plants or nerve gas, right? Sometimes the biological call is coming from inside the house.
Right, exactly.
The classic clinical example of a junction failure in the notes is myasthenia gravis.
Yes, myasthenia gravis. For centuries, medical science was completely baffled by this disease.
Really?
Oh, yeah. Going back to clinical descriptions in the 1600s, patients would present with this profound, fluctuating muscle weakness that got progressively worse throughout the day.
Okay.
And doctors debated for generations.
Yeah.
Is the brain failing? Is the nerve degrading? Is the muscle tissue rotting?
And it turns out it's none of the above. It's a disease of the empty space in between.
Right. But the mechanics of it weren't unraveled until 1934, thanks to a brilliant deduction by a British physician named Mary Walker.
Oh, Mary Walker.
Yeah. She noticed that when she gave her myasthenia patients a drug called physostigmine, their muscle weakness dramatically, temporarily vanished.
Just completely went away.
Temporarily, yes. The genius of Mary Walker's observation was understanding exactly what physostigmine did. It is a mild, reversible acetylcholinesterase inhibitor.
Oh. So it intentionally slows down the cleanup crew.
Exactly. By temporarily crippling the enzyme that destroys acetylcholine, she was leaving the chemical in the synaptic gap just a little bit longer.
Ah, so this increased dwell time gave the chemical a better chance of successfully activating the muscle.
Which was the ultimate clue. If leaving the chemical in the gap longer fixes the weakness, the problem isn't that the nerve isn't firing.
The problem is on the receiving end. The muscle is struggling to hear the message.
Right. And today we know exactly why myasthenia gravis is an autoimmune disease.
So the body is attacking itself.
Yes. For reasons we are still unraveling, the patient's own immune system produces rogue antibodies. These antibodies cross the bloodstream, targets the neuromuscular junction, and physically attack and destroy the nicotinic acetylcholine receptors on the motor end plate.
It literally bulldozes the receiving docs. So the nerve is throwing the exact same number of water balloons, but there's almost nothing left on the other side to catch them.
Precisely. Remember that evolutionary safety factor we talked about?
Right, the fact that a healthy junction over-delivers the chemical payload by 3 to 5 times what is actually required.
Yes. Myasthenia gravis steadily erodes that safety margin. The disease thins out the receptors until the muscle is operating right on the ragged edge of failure.
So when the patient tries to sustain a movement, the depleted receptor pool just can't translate enough of the chemical signal to keep the internal calcium flowing.
And the bridge structurally collapses under load.
Wow. So what does this all mean? We started this deep dive by looking at a completely mundane action, reaching for a cup of coffee, or the granular precision of threading a needle.
Yeah.
And we've uncovered this staggering multi-step chemical relay race that happens in the literal blink of an eye. We've seen how calcium rushing into a nerve triggers a massive dump of neurotransmitters.
We've explored the mathematics of motor units,
Yeah.
trading precision for raw power.
Right, and we've seen how fragile this microscopic gap can be when exposed to botanical toxins, military nerve agents, or our own misguided immune systems. The neuromuscular junction isn't just an empty space between a wire and a motor.
No, it sits at the incredible biological intersection of cholinergic transmission and excitation-contraction coupling.
Which is just a highly active, fiercely maintained environment.
It is, and this raises a really important question, bringing us to a mind-bending detail from the modern definition of the junction in our source material today.
Okay.
For a long time, we viewed the junction as a static anatomical feature. You grow it, and it just sits there.
Like a piece of hardware.
Exactly. But recent molecular research has highlighted the role of complex signaling proteins with names like agrin and musk.
Right. The notes mention that these proteins act like molecular scaffolding. The nerve actually secretes a grin into the gap, and the muscle uses the musk coprotein to receive that signal and densely cluster its receptors right where the nerve is pointing.
Yes, the nerve and the muscle are actively talking to each other, completely independent of the electrical brain signals.
That is wild.
The neuromuscular junction is now understood as a jointly built structure. The nerve and the muscle continuously construct, repair, and maintain this bridge together 24 hours a day throughout your entire life.
And if they stop this chemical dialogue,
The receptors drift apart and the bridge dissolves.
that is something profound to mull over. If our nerves and muscles are engaged in a lifelong active chemical conversation simply to build and maintain the infrastructure of movement,
Yeah.
what happens if medical science finally learns to fluently speak the language of agrin and musk? Could we one day command the body to intentionally lay down new infrastructure, like aggressively rebuilding these bridges in patients suffering from severe spinal cord injuries or debilitating neurodegenerative diseases?
It completely shifts the medical paradigm. Instead of just treating the symptoms of a broken bridge with drugs like physostigmine, we might one day convince the tissue to architect an entirely new connection.
It's an incredible thought. The next time you effortlessly reach out and grab a cup of coffee, take a moment to appreciate the millions of vesicles fusing, the violent rush of calcium and sodium, and the dynamic, jointly maintained chemical bridge that makes every single intentional movement in your life possible. Thank you for joining us on this deep dive.